Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of the show, bringing another really awesome guest for you today, involved in some extremely interesting areas of science and technology, of biomedicine. Today on the show, we have the honor of being joined by Dr. Christopher Buck, who is Senior Investigator in the Laboratory of Cellular Oncology and Head of the Tumor Virus Molecular Biology Section, Center for Cancer Research at the National Cancer Institute. Dr. Buck's lab focuses on the virion proteins of a family of viruses called Polyomaviruses, and their research ranges from basic investigation of the mechanisms through which these viruses infect cells to translational development of various virus-like particles for vaccines. They also maintain really active interest in discovering novel virus species, ultimately with the goal of uncovering human disease associations, understanding the evolutionary history of various cancer-causing viruses. Dr. Buck received his undergraduate bachelor's degree in molecular, cellular, and developmental biology from the University of Colorado at Boulder, went on to do his Ph.D. at Johns Hopkins School of Medicine. His graduate research was in Dr. Robert Siliciano's lab, focused on the translation and immunogenicity of HIV-1 capsid protein. His postdoc training was in the lab of cell oncology, where he developed systems for producing, we actually working with human papillomavirus-based gene transfer vectors. And his work there ranged, again, basic studies of those viruses, morphogenesis related to translational research, identifying candidate topical microbicides for blocking HPV transmission. He's done a lot, and he joined in 2007 the facility at NCI, and ultimately was awarded tenure in 2014. A lot to get into today, a lot of really cool themes. Honored to have him. Dr. Christopher Buck, welcome to the show.
Thanks. Call me Chris, please.
I will, Chris. No, it's great having you. I'm going to start off early on. I want to get back to the beginning here so people can really understand the full arc of your career and where we're going to be going with this discussion. But take us back to, let's say, your dissertation. As mentioned, you worked on HIV and this really interesting gag gene, which ultimately you figured out was an interesting way that HIV used to translate itself, and sort of a different target, let's say, distinct and functional from its traditional viral life cycle. Take us back a little bit. How did this topic become of interest to you, and a little bit of your early history in the HIV space?
So I've been doing vaccines for a long time. It was actually a vaccine project for HIV. We were working on trying to enhance T cell responses, especially cytotoxic T cell, the killer T cell responses in HIV. And you can guess how well that project went. It's 30 years later and we still don't have an HIV vaccine. And I noticed that there were these interesting translation issues. There's actually like an extra capsid protein, and I got interested in how that one is translated. And when I saw a seminar from my eventual postdoctoral mentor, John Schiller, in the research program at Hopkins, I was smitten. I really actually friends in the audience said, "Oh, that guy thinks just like you." And so John was working on the early stages of the HPV vaccine. That one has been successful. And when John came, it was kind of just still a twinkle in his eye. It's now one of the most successful vaccines in human history. It's really great. And the secret of why it works is because it's this repetitively structured capsid. So this is the coat of the virus. And HIV, the capsid, this geometrically regular structure, that's inside of a lipid envelope. And there's this envelope protein that mediates the infection. And for HPV, it's different. It's a non-enveloped virion. And that means it's a naked protein shell, and it's made of one protein. And so that protein self-assembles into this beautiful geometric structure. And the immune system recognizes these structures as grotesquely foreign. It's like a horror movie to a B cell. They see repeat, 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 and there's nothing else like that in the human body, except for the things that detect that type of pattern. Some of the antibodies, some of the elements of the complement system innately recognize these repetitive arrays. And so the HPV vaccine, so successful. You get one dose and you are set for life. You have all the antibodies you need. You'll probably have 100 times more antibodies than are really necessary to fight HPV. And you are protected from the new infection. And this is a key lesson. I call it the Bachmann effect. There was a paper from Bachmann and Zinkernagel in the 1990s. And we've been working, we as a community, the HPV community has been working to try to tell, for example, the HIV people, guess what? We found this amazing thing, and the HIV people keep not getting the memo. It's very frustrating because, it's not easy to take the HIV envelope protein and repetitively array it. But it can be done. And if you can do it, you can make HIV in this same Bachmann category that we have for HPV. And so I would like to see that vaccine. I still have not. And as we move forward, I tell you, this might be the most exciting thing about this yeast approach, where we've got these new food-based approaches, again, using this Bachmann effect. And the fact that they're working is going to enable people to test this hypothesis. We're going to find out whether the Bachmann effect can be reoriented back to HIV. And the part about it being, this story is the story of my entire career. So the part about it being easy means we can do things like look in a person who has HIV, look at the exact sequences in that one person, custom make a yeast that neutralizes the exact variants that that one person has. And we could hope to have an antibody response that would keep the HIV in that person bottled up forever. It would just be enough neutralization that if the HIV tried to come out from its latent reservoir, it would just get fried by antibodies. And I tell you this story, if that works, we could have cured HIV 30 years ago. We could have had a solution that would have meant that people could finally go off of the drug therapies. I could have done it in 1995 when there were not triple cocktails and people were dying. And if only we had thought of this thing I'm doing now 30 years ago. It was just a failure of vision to see that there was this path that was so easy we could do things like custom make a vaccine for a given person.
Sure, it's extremely interesting. We were actually just talking about HIV vaccines a couple episodes ago, and sort of this sort of very unique landscape in terms of mucosal immunity part of it, and then the unique dendritic antigen presentation, and yeah, we went into a lot of that, and obviously we're going to go in a slightly different direction today from HIV into polyomaviruses. But before we get to polyomaviruses, let's stay with papilloma for a moment, because you're mentioning something I'd really like, just if you could say a few more words about, because while you studied these viruses and neutralization and tropism and all the other stuff that came along with it, a very successful vaccine as a result, you learned a lot about these pseudovirus structures, which ultimately we'll be getting into when we get to polyoma. But explain what that is. Explain the principle of what a synthetic viral particle means for people that may be unfamiliar with this term, because we'll be talking about pseudoviruses and capsid a little more later on.
So a big problem with HPVs, all papillomaviruses, is that they're exquisitely tailored to one tissue. They infect your skin, and they only make new virions at the last minute as you're about to shed dead skin cells into the
Mhm.
environment. And what happens is if you take an HPV and you put it into a HeLa cell, any just ordinary single layer culture, it thinks it's at the bottom of the skin and it's not time for it to make new virus particles. And so you cannot culture HPV in a dish. It's absolutely impossible because the life cycle is just keyed to this exotic stage of the differentiation of the skin. So in that case, what we usually resort to is just taking out the guts of HPV and stuffing in a reporter gene like green fluorescent protein or something so that you can look at the delivery of the viral DNA into a new host cell. And if you can do that, then you can ask whether a vaccine recipient has antibodies that neutralize the infectious entry process. And you can do things like discover what receptors the virus is using to get into cells. I also have a fun project from 2007 where I screened a lot of food grade compounds to see if any of them happened to inhibit the infectious entry process. And there's one called carrageenan.
Yep.
Carrageenan is the secret ingredient that stops ice cream from forming ice crystals. It's in every ice cream you ever ate. It
Yeah.
used to not be in Breyers, and I hated Breyers in the 80s because it's so crispy, crunchy ice crystals, but they add the carrageenan now. Anyway, this carrageenan is more like the infectious entry receptor than the actual infectious entry receptor. And people use it for things other than ice cream. In fact, we found out that people were already using carrageenan as the gelling agent in some brands of sexual lubricant product.
Right.
And this
Right.
stuff, like one drop of this sex lube in a bathtub full of water would still neutralize the virus because it's exactly the thing that the virus is looking for to get into cells. So the pseudovirus systems are extremely powerful for viruses you cannot culture. This is not HIV. There are pseudoviruses for HIV, but they're just for like biosafety. In the case of HPV, it's like the old parable of like the in the land of the blind, the one-eyed man is king. So the
Mhm.
pseudoviruses are not perfect. They're not an exact replication of the whole viral life cycle, but they're a lot better than nothing.
Right. Perfect. So that landscape being laid, let's talk about polyomaviruses for a moment. And you'll hear, so this is a distinct virus family from the papillomaviruses. They got some similarities in the sense, circular double-stranded DNA and all that, non-envelope. But these polyomaviruses are much broader tropism, so kidney and brain and immune cells. For the most part, they're harmless, but when things go wrong in immunocompromised patients, potential cancers, and you really, I gotta say, and I'd like you to take a little time with this. You really sort of laid the landscape here because sort of like before you came along, I mean, there was a couple that were identified, and we sort of understood them as sort of an oddity type virus, but you really came along and made a major discovery in understanding how many there were, sort of the degrees of sort of their infectivity. Take us a little bit because before we get again we get to vaccines and beer and all that. Talk a little bit about your role in defining sort of polyoma research field as a separate thing. It's
So
important
what
part of the story.
right around the time that I branched off from my postdoctoral work and moved down the hall from my extremely famous mentors, I joined the tenure track. I was still in the same department, and the top priority in that situation is to prove that you are not just reflecting the glory of your brilliant mentors. And so, about that time, Pat Moore and Yuan Chang, this is the husband and wife team that originally discovered Kaposi sarcoma virus, two of my heroes. They were already two of
Yeah.
my heroes. They discovered the fifth human polyomavirus, and it was called Merkel cell polyomavirus. And that is a rare form of skin cancer. And this polyomavirus definitely lives in the skin. And so I was down the hall from the famous mentors. I saw a skin polyomavirus. The biology is exactly the same. I
Mhm.
didn't have to learn a new virus family, but I could go to to the site visitors and say, "Oh, I work on a different virus family." And so that was kind of the push out of the nest that I got, why I moved to polyomaviruses. In the early days, we actually didn't know what tissues the Merkel cell polyomavirus lived in. The first two, which are what I actually have studied in this vaccine, those are named for patient initials. They're called BK and JC polyomavirus. That's HPYV1 and 2. Those two infect the urinary tract. There was a couple more that were named for institutes. There was Washington University and Karolinska Institute polyomaviruses, and those infected the respiratory tract. This Merkel cell polyomavirus, Merkel cells are a skin cell, so it was a good guess that they were skin. But what we did is we did some of the first basically broad virus hunting in human skin. So we went around the department and we swabbed we gave people a swab to swab their own forehead, and then we extracted the DNA. And then we used this method called rolling circle amplification. It's also known as multiple displacement amplification. And it uses this magical polymerase from phage 529. And it will, when it gets on a circular DNA, it has almost infinite processivity. And that means it goes around the circle. It spins off a single-stranded DNA. It works at room temperature. So you can just add 6 base primers, like just 6 nucleotides that can prime the second strand synthesis. And so you can take any circular DNA you want and enrich it geometrically, essentially, relative to the starting material. So we did that. We basically looked for all of the circular DNA in skin samples, and there was so much. I used basic restriction enzyme-based cloning, like from the 70s, to clone viral genomes. And lo and behold, there was Merkel cell polyomavirus. So those were the first non-tumor wild type healthy skin Merkel cell polyomaviruses. And then there were 2 others, human polyomavirus 6 and 7. And that got me excited. I also have a paper called Hamburger Polyomaviruses. So in his Nobel lecture, Harold Zerhausen, who's the guy that discovered that HPVs cause cancer, Harold tossed out, there may be polyomaviruses or papillomaviruses in raw meat that people eat, and these viruses may be getting into your intestine, and they're not well suited. All these papillomaviruses and polyomaviruses are exquisitely tailored to their host species. So you can't get infected with a bovine polyomavirus or papillomavirus, but it can go on a one-way trip into a cell and make that cell very bad. It basically, the virus is just confused and it's pushing buttons, it's pulling levers. And if that's an early step in cancer, it would mean you probably shouldn't be eating raw beef with this carcinogen in it. It's essentially a one-way carcinogen. And so Harold Zerhausen is great. That's another of my heroes. And I figured, well, this is a testable prediction. So we went to the supermarket and we did the rolling circle amplification on hamburger. And we found several previously unknown bovine polyomaviruses, just as Harold had predicted. So this virus hunting is fun. I'm not doing it right this minute, but when pandemic came and I had to work from home, I stopped going to the supermarket and instead I started plowing through this giant sequence archive called the Sequence Read Archive. So this is like, at the time I started, it was like 25 petabytes of sequence data, and it's all disassembled. And I found ways to assemble it and then go through it at very high throughput to find viruses in other people's data sets. And, so I spent the pandemic at the bottom of the Marianas Trench and in clinical samples that other people have put in the Sequence Read Archive. And that's the way now. I hardly ever sequence anything myself anymore, but there are a lot of viruses out there that people just weren't looking for them. And the moral of that story is actually that if you look at the big picture, these viruses are Lego bricks. We think of polyoma and papilloma, but in fish, there are some ones that are hybrids of polyoma and papilloma. And some of that family also has adenovirus parts. So one of the discoveries was these fish Adomaviruses. This is like adenovirus with either polyoma or papilloma. And those viruses are fun and interesting. This is the destination for the vaccine technology. So there's a condition called blotchy bass syndrome, and sport fishermen catch these bass, largemouth and smallmouth, and they have these big black spots on them, and then you cannot put them on your trophy wall. And you might even feel a little reluctant to eat them. These black spots can progress to cancer in the fish, and it looks like they are caused by adomaviruses. There's there's also one in Japan. It's killing all the eels in Japan, and Japanese people really like eels. And so if you could make a vaccine that would protect eels against the Japanese eel adomavirus, they will make statues to you in town squares in Japan. They will be very happy about it. And so this yeast delivery system, that could work for fish, and we would have to figure out how to employ this Bachman effect. That's actually a hard puzzle. We're working on that for human adenovirus. This is another one of the common cold viruses, and it's not easy. Polyoma, papilloma, it's free and easy. That's why I started with them. But the vaccine technology is married to the virus hunting, because you find the virus and that can tell you, what disease is being caused by what virus. And then you have a vaccine that you can retarget quickly, and you don't have to spend, 15 years figuring out how to purify particles. You can try it quickly and easily in the yeast. So they're married to the virus hunting. Is not a side business. It's kind of also married to the vaccine effort.
Yeah. No, it's important. Let's just say it that way in general,
but mostly it's fun.
Yeah.
I'll tell you the secret to my career is like my operating system. If I'm interested in something, I'm good at it. But steering the spotlight, I very different. I can see that something is important, and I am unable to arouse interest in myself. And so the whole thing just runs on like, what is Chris Buck interested in this morning? And fortunately, like it works, but I cannot claim noble intent. It's just the spotlight is blowing around, and sometimes there's interesting things in there.
No, it's all very interesting, and again, I want to again take some time to go through each part of this puzzle, because it's quite an interesting story. And one thing I would like to spend a little time on also. I mean, so we talked about antigens and capsid structure, but specifically relevant to oral stability, and then all the cool things that happen in the gut associated lymphoid tissue and all that. I mean, talk a little bit about, because clearly for these capsids to survive in the oral environment, structural integrity, pH, enzymes, various chemicals talk a little bit about that part as well, of why these structures make ideal oral delivery systems versus anything else that has been attempted in sort of oral vaccination in the past.
So this part, actually, I was surprised. Our first experiment was to take purified polyomavirus capsid. The protein is called VP1. So if I slip, that's what I'm talking about. So we took purified pseudovirions, like basically just the capsid protein arranged in its geometrical array, and we fed it to mice, and nothing happened. If we put it up the mouse's nose, there were actually quite good responses, and that's not surprising. The nasal vaccine route is kind of a hot topic lately. So for polyoma, this BK and JC polyomavirus, human polyomaviruses 1 and 2, discovered in about 1971. Those two are shed in the urine, and almost everybody is chronically infected with at least one strain. There are different strains of them. And the transmission route is actually not known. We know that they come out in the urine, but then whether these dried urine fomites end up in your respiratory tract or in your mouth or on your skin, we barely know this. And so I always thought it was urine oral, and that seemed like the most reasonable route, that you get like the dried urine on your fingers and then you eat it. So I was surprised to find that the VP1 protein is apparently not at all stable in the stomach. And
Mhm.
I recant. I now think if I were betting, I would bet that BK and JC are transmitted via respiratory and that it's dried urine fomites getting in your nose. So this virus, it has to survive drying. Absolutely. It absolutely has to survive drying. But it apparently nature did not make it to survive the stomach acid. And that's not true. You know, if you're polio, you must survive the stomach acid. If you're cholera, you must survive the stomach acid. And there have actually been a few studies. They're obscure. They coming out of like China and the Philippines and stuff like that. But people have used yeast and have fed yeast vaccines to mice and chickens, and those have all been in the category of flu or COVID, where they take just a piece of the exterior envelope protein of flu or COVID and they display it on the surface of the yeast. And just that piece of flu or COVID displayed on the outside of the yeast, I think that does survive stomach acid. That those vaccines sort of do work at least a little bit. I'm sure we can do better. But the secret was that if you take yeast that have the polyoma capsid VP1 protein inside, this is a non-envelope virus, so it's just all inside the yeast, not displayed on the surface of the yeast. And if you grind up the yeast and feed them to mice, you get nothing. If you give the yeast in a live state to mice, the yeast survive the stomach acid and then
Okay.
they break open in the intestine. So this is known. It's known that yeast only survive for maybe a day at most in the intestine of a mouse. And so eventually the yeast break open, they release the VP1. And then there's another step which is probably important. We're actually right now trying to find out why the polyomavirus vaccine worked. We're taking the coat protein VP1 and breaking it into just pentamers instead of the full virion to ask whether the Bachman effect is important, whether this repetitive array effect is important. But we're also deleting the amino acids that mediate the virus binding to its receptor. So its receptor is a glycan,
Hmm.
it's a cell surface sugar. It's called a ganglioside in most cases. And gangliosides have sialic acid, also known as neuraminic acid. This is the same
Yep.
thing that flu binds, these sialic acids.
Yep.
And the intestinal cell that sort of samples the lumen of the intestine, grabs things, passes them down to lymphoid structures underneath it. This is called a M cell. It's not
Hmm.
multifold, but I think it's called multifold. I don't know what M is for, but these M cells have lots of gangliosides on the surface. And this actually might be a way for some things to get in. So one of the successful oral vaccines that's currently out there is called Dukoral is the brand name. And this is a cholera vaccine. It's
Okay.
killed cholera bacteria plus recombinant cholera toxin B subunit. And CTB
Mhm.
is harmless by itself, but guess what CTB binds? It binds gangliosides, exactly the same spectrum of cell surface receptor that the polyomaviruses are going for. And so if I'm betting that binding to M cells in the intestine is an important thing for the oral vaccine antigens to have. We'll find out. We'll find out. We can do the mutagenic experiment, and with the taking away the binding capacity, we'll find out whether it's still immunogenic or not.
Right. And one other thing, and then we'll get into molecular farming and beer and stuff like that. 'Cause and again, I'm talking outside of my sphere here to smack me down, but the issue. I mean, obviously, when we consume things, we can generate an immunogenic response. Um, But our system currently, I mean, we eat lots of immunogens every day in our food and so forth. Our bodies know, hey, we don't want to mount immune response to all that stuff. And so there's this immunogenicity sort of tolerance issue, right? I mean, um, which, the tolerance and energy and all that is, where the allergy folks hang out with. Can you say a couple words about that as well? Because I know our bodies have to strike a balance there and how you think about we want a vaccine response. We don't want a tolerance response, but we're kind of biased in certain ways. And now I'll shut up and I'll let you say
Yeah,
something about that.
this was the dogma that stopped me from doing the experiment in 1996.
Okay.
So my buddies and I in grad school, we talked over beers about whether we could make a beer vaccine or not. And we all thought,
Right.
well, we all know that you're not immune to everything you eat, and
Right.
it's probably not going to work. And this defeatist attitude is the problem. And we do know that some oral immunogens work. Certainly, polio vaccine, which comes on a sugar
Sure.
cube, that works but probably because the virus is infectious and it infects the epithelium of the intestine and then replicates and makes more of itself, and then that's displayed to the immune system. So yeah, these oral immunogens, the dogma has always been that we will either ignore them or it will trigger tolerance, where you can try to cure yourselves of allergens by eating them. Apparently, like the Native Americans apparently had the habit of eating poison ivy so that
Yeah.
they didn't get it on their skin in the woods, but it was horrible. Like they had bloody diarrhea for a year
Mm.
to achieve this. And it's true that certainly this is what we would expect, is that you would tolerate the things you eat because you really don't want to be reacting to everything you eat. This is where those M cells, this is why I like the M cell idea. It may be that things that are engineered to specifically bind to a particular antigen presentation cell in the intestine are treated differently than some random phage that has no way of interacting with a human cell.
Got it. Got it. Okay. Yeah. Again, I didn't want to mess with dogma, but I know I understand where you're going, and no, that's perfect. That's fine. Again, I wanted just to lay that out there for people that had that question, and thank you for clearing that up. Now, this being said, um, molecular farming as an area, it's a place I hung out a couple careers ago. We've had some successes in the sense that right, I mean, they made ZMapp for Ebola a couple years ago, made in tobacco. BCG vaccine, I believe, is still produced on potatoes. There's a lot of examples. It didn't become a major multi-billion-dollar industry, but there's some good success stories out there. Talk about the beer strategy, how you came up with it. You're hanging around drinking some beers, talking about beer, vaccines. Well, give us a little of the background where you sort of put the four things that we've talked about now: capsid, pseudo antigens, oral root. Was the fourth thing? Whatever, there's four things in there. Okay. Let's make this thing in yeast and try to produce the world's first beer vaccine.
I mean, I love these food vaccine ideas. Like but what I know now is that if I took Polyoma VP1 and I put it in a tomato, I don't think it would work because I think it would all just die in the stomach acid. And so having it inside the live yeast that can protect it against the stomach acid, that was the secret of the experiment. And it came from trying to do the normal thing the right way, which is we've been making a traditional injection vaccine with purified Polyoma VP1 coat protein. It's for 15 years we've been pursuing that. We licensed the patents we have in that area to an Indian company called Biological E. I guess
Hmm.
disclosure, I get
Yep.
some licensing money goes into my bank account from time to time.
Yep.
Although if I were only motivated by money, I don't think I would be telling you how to make free vaccine in your kitchen.
Right.
So I think
Right.
the money is not a big part of my thinking about it. Anyway, BioE licensed our patents in 2019. They got distracted because they were one of the biggest companies making COVID vaccines. And so
Hmm.
they had a couple of years where they had other
Sure.
things they needed to do. But we're coming up on 6 years later and they're still working on, I'm not allowed to tell you what they're doing, but you can find out for yourself. They're not in clinical trials right now
Yeah,
anywhere in the world. So you can tell like they're still working on the stuff that goes in the syringe and getting such stuff past the regulators. And so in service of our licensee BioE, we started playing with expression in yeast. We had originally
Hmm.
done insect cell systems. These are a little more challenging, but sometimes the quality of the particles is better. But we had, actually what happened was we had a Fulbright scholar from Lithuania. She came, this is Amelia Vasilianaitė. She came from Alma Gedvalaitė's lab. Alma is in Lithuania, and she is the world's top expert on making polyoma coat proteins in yeast. And so Amelia kind of came with these yeast. We were actually working on an approach using yogurt first. So the idea is you take yogurt bacteria, you find a bacteriophage that infects the yogurt bacteria, and then you engineer the bacteriophage to force the bacterium to make lots and lots of VP1. You can make reasonable quality virus-like particles in bacteria. But anyway, engineering the phage was hard. We're still doing it. We're still working on that. And it might work. That might actually be the way of the future. But meantime, Amelia on this, international exchange program just brought the yeast, and then we started playing with the yeast. And it didn't take long before I thought, like, what happens if we just feed this yeast to the mice? I've been a home brewer since the mid-90s. And so I thought, if you can just eat it, like, that's something I know I can do in my kitchen. I have done this sort of thing before. And so we started with the right experiments where we did the purified antigen and we did the ground up yeast. And it took some effort to convince the animal facility that, like, just pouring some yeast on mouse kibble was a good experiment. Everybody thought, oh no, you have to find a manufacturer to do a controlled formulation. And we have to do gavage. That's what they said, is you have to snake a tube down the mouse's throat so that you can do a controlled dose. And I said, like, I'm not doing any of these things in my kitchen. I just want to find out what happens if you eat it. And the first experiment, I have flashbulb memory of where I was standing when I saw the 2-week data, and it was just the barest whiff of a response at 2 weeks after the mice ate the live yeast. And I saw it, and, like, I literally felt weak in the knees, like earthquake is going on, because I could see the whole path laid out ahead, that if live yeast, which are a food, can immunize, then vaccines can be a food. And if vaccines can be a food, that can be regulated as a food, and that it just changes everything. The idea that a food can serve as a vaccine, it's transformational. I could see this whole, we're still on the path, but, like, I can see what the path will look like in the future. It will be a revolution.
Mm-hmm. And you focused on, it was the BK type 4, I believe, in your own experiments, or talk about what you did in terms of your biohacking experiments, your home brew, because again, I want to hear a little bit about that, some of what you learned, and then we'll go a little further in terms, because as you're pointing out, obviously it's a very good production platform and a delivery platform, but again, the diversity. Clearly, you've discovered a lot of these, and the potential for orally delivered vaccines that taste good, that potentially hit a wide range of these viruses simultaneously, I think is extremely interesting due to the connections to certain cancers.
The first thing I did, I again did the right thing, and I told the institutional review board that I wanted to make beer and drink it. And we hit the skids. The IRB, it didn't even get to the IRB. There was first an institutional bioethics committee that needs to be consulted before the official IRB can even consider the proposal. And the institutional bioethics committee wouldn't even let me submit the IRB proposal, because they said nobody can ever do any self-experiments. I actually think they have misread the law with this opinion. But they said that before I could work with the yeast, I needed to first submit an FDA investigational new drug application. And this is familiar. This is the way that we will make the actual vaccine that we're working with BioE. And this is the familiar way that everybody at the National Cancer Institute thinks is the only way we can do things. And we just don't ever think about food. I mean, I think about food. I thought about carrageenan way back, but nobody has thought about using food for health benefits, which I find regrettable. And nobody understands food law, which is forgivable. We are not lawyers, but I read the food laws and I told the bioethics committee that they had not read the food laws, and here's what the food law says. And one insidious thing, and I call it insidious, I think this is a very bad bit of law, but the law says that if a new product first appears as an investigational new drug, it can never be a food after that. It always has to be a drug, and it can never be a food. The reverse is not true. If you have a food
Right.
and you want to turn it into a drug, you can definitely do that, and you can make it an investigational new drug, but it can still be a food on the side. And so an example of,
Unless you pull it off the market. You could actually, sorry, I didn't mean to interrupt, but I was in this business at one point. You pull it off the market and say it's no longer déchet, and no one else can follow you after that. Bad, sorry.
yes, if you have dollar signs in your eyes, that's what you
Right.
should definitely do. But there, I mean, there's examples like the malaria drug artemisinin. It
Sure.
comes from sweet wormwood. It's one of the ancient Chinese remedies for malaria. There is a dietary supplement product that is on the déchet market. It's
Yeah.
called artemisinin. And so the fact that artemisinin is an FDA approved drug does not stop it from also being a food or food supplement product. And so, in other words, the bioethics committee said when they told me I had to submit an IND, I heard it as, well, that's a beautiful baby you've discovered. Why don't you smother it to death and then we can all study it? And the IND was just not an available option. I could not comply with the misreading of food law that I thought that the bioethics committee had read. And what I could read was that there is nothing stopping me from making the thing in my own kitchen. And so actually the GMO laws have changed in the last 6 or 7 years. Most people don't realize it, but there are a lot of commercial brewers yeast that are GMO products now. And so they have been generally recognized as safe products, and that's it. And you can self-affirm generally recognized as safe. So
Yeah.
one example is there's a yeast that is engineered to secrete lactic acid, and it makes the beer tangy. Ordinarily, you would sour it with like some yogurt bacteria, lactobacilli, but then the lactobacilli got everywhere, and breweries don't like having these bacteria around. So they prefer, some breweries prefer to use this engineered yeast. Half of the breweries in my local area are using GMO yeast, and half of the ones that are using it do not know that it's GMO. So not only are they not telling customers, they're not even aware that what they're using is technically a GMO organism. This is all very different in Europe. In Europe, it would be illegal to do what I did in my kitchen, because the GMO, even to make a GMO, you have to get institutional permission or regulatory permission. But in the US, it is Wild West now, and you can make whatever GMO yeast you want, and you can sell it so long as you have a clear case for why it is generally recognized as safe. And in the case of this BK polyomavirus, I told you everybody's shedding it in their urine all the time. And so it's a thing it's already in the environment. It's already in your diet. I mean, I don't like to think about drinking urine, but like people have done that in human history. And so this particular protein that I'm making is already a common known component of the human diet, and it is not known to be harmful. And that is classic textbook, generally recognized as safe. If you find something people have been eating for centuries and you want to put it in your new product, you are allowed to do that under a self-affirmation of generally recognized as safe. And so the conflict with the bioethics committee was solved by me reading the law as a US citizen. I founded a small, it's not just nonprofit, but it is non-equity. It's called Gusteau Research Corporation. This is for the chef in the movie Ratatouille. And so the chef's catchphrase was anyone can cook. And so I founded this little company. I bought some basic molecular biology equipment. I made the expression plasmid that I needed to transform into the yeast in my kitchen. This is possible to do that. There's now DNA synthesis companies, and this was part of the reason I needed the Gusteau Research Corporation, is because the DNA synthesis companies probably won't sell to one guy in his basement. And the process was reasonably straightforward, and I accomplished it all in my own finances, all in my free time. And all of that is perfectly legal. And then I drank the beer, and that is perfectly legal as well. There was some more recent controversy for the process of testing my own blood to find out whether my antibodies had gone up or down. So this would address the tolerance question. If my antibodies go down, that would be I would stop drinking the beer. Basically, that would be a sign that the tolerance is being induced, and that would be a disaster. That would be dangerous. It would be dangerous because it would reduce my immunity to a known pathogen. So it turns out this is not the first time I thought about these self-experiments. So there was a time about 10 or 12 years ago where I just wanted to do some hemagglutination assays. So this is the classic virology assay where you look for a virus to glue together red blood cells, and then you can see that they don't drop to the bottom of a round bottom well. It's an easy classic assay. And I just want to do a hemagglutination assay on a few samples, and I don't want to have to buy like a whole bag of blood just to get some red blood cells. I got red blood cells in my finger. And so I actually have an IRB approved protocol that authorizes me to prick my own finger. I don't think I should have had to ask for permission to prick my own finger, because it's obviously safer for me to use a few drops of my own blood than it is for me to use somebody else's blood that I bought on the open market. And so I just don't see how the IRB could possibly tell me that I am not offering informed consent to myself, or that I'm endangering anybody. I'm making myself safer. And I thought this paperwork was absurd and stupid, but it's still on the books. So I drank beer in my kitchen on my own power, but I am authorized to prick my own finger and test my own blood in serological assays. And so I just availed myself of that. But the administration, the leadership is still uncomfortable because they are conflating my self-serology, which is authorized, with the unauthorized kitchen experiment. And I'm mad about that view, because I don't think they have any business telling me what to do in my kitchen. But that is the premise of the entire argument, that the IRB, or the bioethics committee that wouldn't let me get to the IRB, thinks that they can tell me that because something is related to my work, that I need their permission to do it. And I find it unethical. I find this judgment completely unacceptable, and I'm willing to fight about it. I think somebody should fight about it, because I think that the infrastructure has actually lost sight of the law. This law was a response to the Tuskegee study. And what happened in the Tuskegee study was an institution told people they couldn't have some medicine. And they're essentially, they're not protecting everybody against the Tuskegee study. They're repeating the Tuskegee study when they tell people they can't have medicine. That was the problem. And they're repeating the history rather than guarding against it.
Yeah, it's always shocking when you hear stories like this, and then you look at whatever, I don't know if all this peptide stuff that's going on nowadays, and entirely illegal, but just turning the blind eye to all of it, all this experimental stuff that's out there that it falls in such a weird gray zone, and people are injecting all sorts of stuff, but you can't drink some beer.
But I would dispute entirely illegal. What I've learned about all this food stuff is that things only become drugs if the manufacturer has label claims. So the law, the actual word in the law is manufacturer intent. If you have the intent to treat disease, then it becomes a drug. But the intent can only be, the FDA is not a mind reader, so the intent can only really be inferred by what's printed on the label. So when these Chinese peptide manufacturers are sending these peptides, I mean, they know what it's good for, but how can you know their intent? You can't know their intent. Here would be an example. If you have vitamin A deficiency, your doctor can tell you might want to eat some carrots, right? The grocer cannot say this carrot is intended to treat your vitamin A deficiency.
Right.
You can go and buy a carrot with you can have the intent of treating your own vitamin A deficiency by buying a carrot, but the key thing is just what the stated label claim is. That's
Right.
really what actually gets regulated. So I'm disputing your claim wholly illegal.
Right.
Only
Okay.
becomes illegal when you say that
Right.
that's what you're doing.
You're right. You're right. No, I stand corrected on that one. But again, Barry Marshall got the Nobel Prize for drinking H. pylori in his kitchen. So yeah, um, very excited and interested in where you're going with it. And
Barry
yeah,
Marshall had the insight that he shouldn't ask.
right,
He's
right,
on
right,
video
right,
saying like it's better to ask forgiveness than
right.
permission. I apparently did not learn Barry Marshall's lesson when I asked the IRB, but
You know, it's interesting. Well, Chris, it's you were talking before about the eel story, and it got me thinking because I was looking at several of your papers here where you talk about sort of the gene transfer among some of these different viruses between different animals and all that. And it just got me thinking of you know this One Health team that we spent a lot of time on. We just Ellen de Barbander, who heads up research at Elanco, which is the third or fourth largest animal vaccine company in the world. I mean, there's a lot of animals out there that have all sorts of interesting viruses that we don't hear that much about. I mean, these are major, when they screw with the cows and the horses and the pigs and all that, these are multi-billion dollar segments. Interesting on that front. I mean, obviously you talked about feeding stuff to animals and then obviously feeding stuff to you, but any intermediary projects there, you play around with cows or pigs or whatever the case may be that you could further expand this principle because I think it has a lot of merit.
it there's like a good news story from the virus hunting, which is that like I don't think there are major new families of viruses. I think there will be new variants of known
Right.
viruses, and there will be some chimeras. I think there will be some ad-oma type things. But even there, what's happening? I'm in the final stages of making a searchable database. So there's
Okay.
basically you watch the TV show Silicon Valley, where like
Sure.
the secret is compression, and I've essentially figured out how to compress petabytes worth of sequence read archive into a searchable database. And where you can ask, here is a protein sequence, which sequence read archive datasets is the protein sequence in? And by searching by protein sequence, that's the key, because that's what nature conserves. It's not conserving the DNA nucleotide sequence.
Hmm.
And so I'm getting to the place where I got the parts list. Like I think I certainly know all of the replicases, all of the things that polymerize nucleotides, all the things that torque apart the DNA so that the host cell machinery can help replicate it. I can certainly find every large tumor antigen. That's the thing that replicates polyoma. I can certainly find all of those at this point. I can find many, many categories of major capsid protein. And I don't think there's going to be much left. And this is actually the joy of all this chimerization, is that there can be some exotic new replicase, but eventually it's going to recombine with a recognizable coat protein or vice
Hmm.
versa. And so if you have one thing, you can find all of the other things, because eventually you're going to find it in this like Lego puzzle where it will be married. The thing that you know will be married to something that you cannot identify, and that will help shine the light on something that would otherwise not be identifiable. But the key parts, the replicases, I can see those clear back to archaea. Like those are conserved enough that I've got the basic parts list, and there's not a lot of universe left to explore in these viruses. I think we're coming to the end of the parts list. So and the Lego brick, this like horizontal recombination, we're talking tens of millions of years. So this is not happening in real time. This is not happening on the fly. These adenoviruses are probably 100 million years old, and there's not a lot of that monkey business. It's rare for these big families to make babies together. That's not a common thing.
Comes next? I mean, again, I love the principle of what you did. Clearly, um, you thought through all the pieces of the puzzle in terms of, creating a stable, early bioavailable novel vaccine candidate here. You know, eh, Again, if the clouds opened up and $10 billion fell into your bank account, I mean, what would you do? I mean, where were you going with all this? I mean, what do you want to develop? What do you want to, where are you thinking the future of edible, well, edible vaccines in general? Because as you pointed out, it's been on the drawing board for a long period of time, with some small wins along the way. But where are we headed?
Well, I don't. You might have already talked to Preston Estep. Is that name familiar? He had early in the pandemic. He founded RadVac. I forgot what that stands for, but they had a peptide-based nasal vaccine that they self-experimented with. And RadVac is still around. I admired them early pandemic, and then I lost track of them. But RadVac is already they are already putting this year's seasonal flu strain into yeast. And so this year's substrain K is going bananas because the current vaccine that you can get is not well matched. And they can put exactly that thing, and a few weeks later, you can be drinking yeast in principle. And so
Nice.
that's good. The biggest joy of all this is that yeast, when in the brewing and distilling industry, it's mostly thrown down the drain. It's mostly just discarded. In this distilling industry, it's a little different. So when you make bourbon, you put the grains into the fermenter with the yeast. It all ferments. It makes a strong beer, basically. And then you take the strong beer, you distill it, but the spent yeast and the spent grains, those distillers routinely feed them to farm animals. In fact, it's a partnership because the farmer comes and picks up the grains, and then the distiller doesn't have to haul them to the dump. But it's free. If you put a bird flu hemagglutinin into the yeast that they're using to make the young bourbon, the strong beer, then that yeast is there. It's live. It falls out of suspension alive, and you could feed it to chickens, and then chickens get vaccinated against bird flu, and then the price of eggs goes down, and then they can never fire you because you're that guy that brought down the price of eggs, and then you're bulletproof against all political considerations. So that's a dream. But I missed a question. So you asked why BK genotype 4, and that sounds like an esoteric question, but the simple answer is because I didn't have any antibodies to it to begin with. So I chose that one so that I could see myself go from nothing to something. There are 4 genotypes, and I had antibodies to BK genotype 1, a little bit of antibodies to 2, and 2 and 4 are sort of cross-reactive a little bit. And so what happened to me was my BK genotype 4 antibodies came up, but also my BK genotype 2 antibodies came up even more because that was essentially a recall response. My immune system was remembering a thing it had already seen.
I see.
And so the reason these viruses are important, the thing that biological E will be targeting initially is a specific application, and that is organ transplant. So if you're like me and you have no anti-BK genotype 4 neutralizing antibodies, and you get a kidney that has BK genotype 4 in it, the kidney comes into you, you go on immunosuppression so that your T cells don't reject the organ, and then the virus goes nuts and there's no neutralizing antibodies to stop it. So if you're on the organ transplant wait list, which is typically at least 6 months, you have to wait a while to get the organ usually. If you're on the organ transplant wait list and you drink the beer vaccine, you get these antibodies. My antibodies are already flat for 6 months. This is already looking like the Bachman effect, just like HPV, where the antibodies come up and then they're flat forever. It's not like the COVID vaccine where it's, coming back down after a few months. And so the organ transplant wait list candidate could drink the beer, get antibodies, and then the antibodies will neutralize any BK4 that happens to be in the kidney. And we can quibble about how much neutralizing antibodies you need. For HPV, you have to protect these stem cells, these keratinocyte stem cells in the
Hmm.
skin, they are bathed in serum, and you need a neutralizing titer of 10. And 10 is like nothing, it's almost undetectably low. But for polyomavirus, this BK polyomavirus, it's infecting the inner lining of the bladder. And in your urine, you're shedding about 1/500th or 1/1000th as much antibody as is in your serum. And so to protect the inside of the bladder, you need 1,000 times more antibodies. And my neutralizing titer was not quite 10,000. 10,000 is rock star. For COVID, it's somewhere in between. You generally have sterilizing immunity at a neutralizing titer of 1,000. So I didn't get to 10,000 for BK genotype 4. I did get to 10,000 for BK genotype 2. And I could probably get more doses or snort some and probably get my titers up. And it may be that having some T cells around that are in a sort of an active state would protect me, even though my antibodies may not be quite enough to get sterilizing immunity against the BK genotype 4. So that's all great for organ transplant recipients, but guess what? That is not the only thing that BK does. You called it harmless before, and I gritted my teeth. So harmless is an unsupported claim in my book.
Okay.
I think they could be doing all sorts of harm, and we're just not smart enough to figure it out yet. I think this about every virus in the universe. I think if you see it and it's there, it's probably doing something bad, and it may be very hard to figure out what the bad thing is. So one example is a common condition called painful bladder syndrome. It's also known as interstitial cystitis, and it's a few percent of older women. It kind of comes and goes, but they have pelvic pain and horrible urinary urgency where they feel like they have to pee every 15 minutes, and it's really debilitating. We don't know what causes it. It's not just a bladder infection. It's not bacterial. But what we do know, there's a half a dozen papers that show that a lot of women with painful bladder syndrome shed a lot of these BK or JC polyomaviruses. And we know they're there. We know that some organ transplant patients get a bladder condition called hemorrhagic cystitis. This means you're bleeding out of your bladder, and it can be lethal, but it's incredibly painful. And so painful bladder syndrome, the solution to the causality problem is not Koch's postulates, because it's impossible to do Koch's postulates for these viruses. They won't infect an animal, so you can't do the experiment. Right. The causality experiment is drink BKJC vaccine beer, and if the pain goes away, that virus was the cause, and there's just no way around it. And so that experiment is incredibly exciting. This is not the big picture long-term where I'm bringing down the price of eggs. This is like next month. Next month, somebody with painful bladder syndrome can drink this beer. And that's what we're going toward at this point. Beyond that, there was this great paper in Science just back in December by some friends of mine that look at this innate antiviral defense. It's a whole system called apobecs. So the apobecs are mutagens. They take amine groups off of cytosines and convert the cytosine to uracil, and that's a mutation in the DNA. Okay. And the system uses it to fight the nucleic acids of viruses, but it's a blunderbuss, and it hits the cellular DNA a lot. And so we know that this polyomavirus infection can activate apobecs. And we know that bladder cancer has a ton of apobec damage. And so what this science paper showed is that when you have a cell infected with this BK polyomavirus, the infection triggers an interferon response, and interferon gamma signaling activates apobec in a neighbor cell. And so it's called transmutagenesis, where the virus is infecting one cell and causing mutations in the neighbor cells. And BK polyomavirus could cause every case of human bladder cancer in the history of the world. That's possible. And that's a long experiment. It will take 30 years to find out, after a bunch of people drink the beer, how much less bladder cancer we get. But when I talked about fighting with the bioethics committee, I wanted to drink this beer because I don't like bladder cancer. I'm not
Right.
planning to get a kidney transplant, but I'm a human being who doesn't want bladder cancer. And the risk of the beer, I guessed correctly, was essentially zero. There are no detectable side effects. And if it can give me some protection against these viruses, guess where else BK shows up? Brain disease. So its
Yeah.
neighbor, JC Polyomavirus, is famous. About 10% of people with AIDS get this condition called progressive multifocal leukoencephalopathy. This is
Mm.
many sites of infection in the white matter of the brain. And it's a brain disease that is lethal to AIDS patients. It's also a side effect of some of the immunosuppressive therapies, like for MS. Some of them come with the side effect of this PML, progressive multifocal leukoencephalopathy. And I don't like that disease, but like, what if there's a little PML in the
Right.
healthy population? What if that thing is like the cause of 10% of all the Alzheimer's in the world? This is
Right.
actually possible. I know it's in the brain. I know this cannot be good news for me. And so drinking this vaccine beer, it's not different from penicillin for syphilis patients in its Tuskegee study. Like denying people the right to choose medicine is very dangerous. And we
Yeah.
just cannot do it lightly as we have been doing.
Yeah, and with the recent, shingles vaccine data and dementia, I mean clearly uh
Exactly. That
we
that study was a Tuskegee, an accidental Tuskegee abuse. Like when they
yep,
developed this age cutoff, like that was abusive. They screwed some people. Some people got
yeah,
dementia because of an arbitrary age cutoff where they were denied a medicine that turned out to be life-saving. Nobody had
yeah,
any way of knowing it, but that's not the point. The point is
right,
that
right,
any medicine that people want that could be life-saving, that the choice should be the patient and not the institution.
right. So I ask about one other thing while we have you today, because in sort of your many responsibilities per your NIH website. I mean there's this human microbiome in cancer sort of subgroup. And I was just interested because we a couple of years ago we did an episode on sort of the beneficial viruses, right? The ones that, hey, if we have this virus X, it seems like these other diseases don't aren't as infectious or they go away. And I forget it was a hepatitis G. I think we talked a little bit about stuff like that. Anything interesting happening in terms of the beneficial side of the virome that you're working on, or is that not part of your? Because obviously you run into lots of these viruses in your studies, and not all of them are bad. And anything interesting popping up there related to cancer,
I mean, so
or other disease?
we're all familiar with the bacterial microbiome, and like a classic question is if I had a magic vaccine that could protect a baby against all the papillomaviruses and polyomaviruses in the universe, would I give that to the baby? And there's like a fun story. My buddy Denise Galloway at University of Washington um, has shown that some of the skin papillomaviruses, they're not the sexually transmitted cervical cancer ones, but what they may cause some amount of skin cancer.
Yeah.
Those ones have the effect of preventing cells from dying after they've been exposed to ultraviolet. So it helps the cell decide to live. And so Denise's joke is like, maybe if you had no papillomaviruses and you got a sunburn, all your skin would fall off. I don't, she's joking. I don't think this is true. I think a vertebrate can live just fine without papillomaviruses. But I, I'm a betting man. No, I think we're better off without them. And I know bacteria are doing me favors. And there's another story. Do you know this guy Skip Virgin? He was in Wash U. He,
It's familiar.
he's mostly famous for training a whole bunch of like Ken Cadwell is one of his trainees, and so Skip had a paper where he showed that herpes viruses can protect mice against other challenges like Yersinia. And what the herpes virus is doing is it's in its latent reservoir and it's periodically waking up and saying like, hey, anybody out there, right? And then the T cells are like, oh God, there's something, right? And so what he postulates is that having this latent herpes infection is just tickling your T cells and just keeping your T cells awake so that your immune system is kind of more active than it would be if you didn't have the herpes virus. And so maybe that's a favorite. Maybe that's the thing that you're talking about. And
Yep.
maybe the baby would be sad if it had no polyoma or papilloma, but maybe the baby would not be at all sad. And if I'm a betting man, I would bet on like we're just better off without them, and they're probably not doing us any favors. Oh, I have a great story. This is a fun story. So it's I can make this vaccine and it's easy. And why do we put up with these viruses if they're so easy to get rid of? They've got this Bachman effect. The vertebrates should have been able to get rid of them. And so it does smell like they're doing us some kind of favor, like we're putting up with them because they're paying their rent in some way. And so here's the way I want them to be paying their rent. It turns out both polyoma and papilloma, both of which are, some of the flavors are in your genital tract, both of them are very sloppy at taking up the viral DNA. This is why they were easy to make as pseudoviruses, because they're kind of like, they'll take up anything. We don't know what the packaging signal is. It may be the spacing of histones or some kind of like meta signal that's not exactly encoded in the sequence at a linear level. But they will take up lots of cellular DNA. So if you look at the virus particles that come out of an HPV skin wart, about a quarter of them have cellular DNA inside of them. And it's just a headful. It's just, 8 kb of cellular DNA. And so here's the partnership. The partnership would be in your genome, things that you want extra copies of, like olfactory receptors. You've got a couple of thousand olfactory receptors, if you count all the dead ones that we lost when we stopped smelling things. What you would do is you would make the packaging preferences for olfactory receptors match what HPV likes to take up. And then HPV takes up an olfactory receptor and delivers it to an embryo, right? And then the embryo or to a sperm cell, and the embryo or sperm cell gets an extra copy of that gene that is delivered to the cell nucleus, then it splices into the genome, and then you've got an extra copy. And so they would be basically handmaidens of genomic plasticity. They would just be a way of enriching for genes you might need more copies of. Right. And so if a lineage completely lost polyoma and papilloma, it's stuck with the 2000 olfactory receptors and it's never going to get this duplication event. And then your lineage just gets snuffed out. And that's a service I can live without. I don't need my genome scrambled anymore. Over 1 million years, it was good for my species. But for me personally, no, I don't need this service anymore. And so my hope is that that's the kind of service that they've been rendering to us. And it's something that the baby will not be sad about.
That's cool. No, again, I just wanted to cover everything that you work on. So no, I appreciate that insight. What's coming up? What else should we know about as we get into 2026? Any other new projects, new viruses that outside of papilloma and polyoma that you're working on? Anything else happening down at NCI that's hot, that's public facing while we have you today? And then anything I missed, please.
So we are working on flu, COVID, and adeno. Adenovirus is a fun story. So when you join the military, there's a vaccine for adenoviruses type 4 and 7. When you get a bunch of 20-year-olds in boot camp and they're all stressed out, adenovirus can sweep through the population. It kills people and it knocks them on their ass for a couple of weeks. And so what they do is they give a vaccine to everybody when they join basic training. And the vaccine is full strength wild type adeno 4 and adeno 7. And the vaccine, you drink it, and by drinking it instead of getting it in a respiratory route, the infection is much more manageable. The immune system can see it and clear it fast enough for it to work. And it's uncomfortable. People get diarrhea and upset stomach, and people do not love this vaccine. But it's a challenge experiment, right?
Hmm.
And so the experiment would be make yeast vaccine, recombinant yeast vaccine, and find out if it prevents people from having symptoms after they
Hmm.
get the Ad 4, Ad 7 challenge. And by the way, it might be come in the form of beer, and
Hmm.
then there will be statues to you in military towns across the country because all the
Yeah.
recruits will be happy that they're drinking beer to protect themselves against the vaccine challenge that they're kind of forced to take.
Right.
So those ones are on deck. What I'm actually doing right now is trying to convince the big yeast supply companies to start making products that people can play with. This will open the door to, I mean, anybody can do this. It should not be my lab doing this. There should be 1,000 other labs working with viruses that they know and like already. And that's the future is not what I'm doing next. But in order for that to happen, we'll need systems for people to share plasmids and systems for people to buy yeast and systems for home brewers to be able to buy the yeast and brew up, a new batch, and places where a woman with painful bladder syndrome could just buy the yeast and chug it and not have to learn home brewing, and systems where distillers and microbreweries could get in on the action, and it could become a whole ecosystem, and it works on capitalism. I am a capitalist. I call myself a capitalist, but when I say it, I mean what Adam Smith described capitalism as, and capitalism requires that there be competition. If you have monopoly, this is not capitalism. And if you have patents, it's also not capitalism. And so the system is hard because we're relying on these monopoly powers and these windfall profits. And if we can just get a system where people can sell stuff and then Consumer Reports can give an objective third-party evaluation of it, like the whole thing is just, it will be a miraculous like feat of capitalism that will work much better than this system where you have to spend 15 years self-testing your own product and convincing the FDA that you self-tested your, like, think if we had this for cars.
Right.
What if you could only buy a car after GM tested it and then the government told you that's the only car you can buy? Like nobody would put up with this for any other product except for medicines. And the food-based approach just gets us out of that entire framework. And it's, you know what, not just vaccines. People have put GLP-1 into yeast and
Yeah.
it kind of works. I wouldn't take it because I'm a hedonist and I don't, and I'm not obese. And I would want to be careful. I don't, the GLP-1 yeast probably couldn't qualify for automatic, generally recognized as safe food status. Uh, but you could make them. People could
Yeah.
do that
Sure.
right this minute. They could homebrew such a thing in their kitchen, as I did with this thing. And so it's not just vaccines. There might be other stuff. I won't want to do it for cancer drugs or anything like that. But like there may be other niches where the food-based medicine approach could be feasible, and it will be equally revolutionary for the capitalism reasons that I'm on about. Like it will just make everything better if we can have a market force that would manipulate this stuff.
I think it's an exciting story. And I've always personally been passionate about sort of this intersection between food and health. And obviously, the last 100 years or so, we forget that a large percentage of all our medicines have come from nature in one
Yeah.
form or another. So it's, you're reconnecting these teams. And I think that's exciting. And, kudos to you for taking on some of these challenges in this very unique way. But it's really impressive. I wish you the best with it, and as well as all the work you're doing at NCI. But again, for everybody out there that's going to be listening to this episode, across the various podcast networks or who will be watching on the YouTube channel. You've been spending time with Dr. Chris Buck, Senior Investigator, Laboratory of Cellular Oncology, Head of the Tumor Virus Microbiology Section, Center for Cancer Research, National Cancer Institute. Chris, again, I want to thank you for taking the time today to walk us through this really exciting story. Thank you for what you do to test the waters and these different boundaries. And we'd like to say here on our show, thanks for helping to create a better tomorrow, particularly for patients via what you do. Really, really interesting. I look forward to continue to follow you.
I'm super grateful for the attention. Thank you so much for having me. Your attention is just as important as the actual work. Like if I'm just one guy and nobody knows about it, this is not a good world.
We'll help you get the word out there.
I have one other little topic that you can edit in or not
Please.
as you wish, but I believe that all scientists should have science blogs. I think actually this is an obligation for us to talk to the public at this point in human history. And I got in on that action again with food. So there were some papers early pandemic that showed that if you feed hamsters mint tea, they get better faster. And there were a lot of other, they started from the Chinese medicine canon and
Right.
just looked at what worked. There are other food products that work. There are other retargeted drugs. And I think it all actually hangs together under like an immune skewing framework, not so much antiviral directly, but just like pushing the immune system in a better direction. All that stuff is the first 10 posts on my Substack blog. And that was what kind of got me into it. But yes, I'm echoing again what you say about food. I think we don't think about the utility of food enough. We're too quick to dismiss it as all snake oil, which, by the way, did
Right.
you know snake oil, the first products actually probably did work. They had a whole bunch of like, omega-3 fatty acids, and they probably were actually effective. And things hit the fan when they started substituting not snake oil for other
Right.
products.
Right. No, it's funny what you were just mentioning before because I have your paper here on COVID and mint. So,
Yeah.
yeah, again, yeah, it's full circle, and yeah, I will put the link to your blog as well, so everyone can check that out.
Okay. Yes,
Aside from the fans, yeah. So
everybody must do this. There should be peer-to-peer review in this form, and science would work better if scientists were talking to each other and the public in a way that everybody could comment. Like things just
um,
go better if we do it that way.
absolutely, absolutely. Great having you, Chris.
Okay, great.
Thanks so much.
Thank you.